Okay, welcome back for lesson number 4, entitled Inferring Genomic Relatedness and Understanding Genomic Results. My name is Jenny Crain. I'm a genomic epidemiologist at the Virginia Department of Health. So this is the last lesson in this series of 4, and our section goals today are to understand when to use differences in alleles versus SNP differences, to restate what inferences a SNP matrix can and cannot inform, identify basic components of phylogenetic trees, to learn some methods used in building phylogenetic trees, and discuss tree building considerations, as well as to gain some general exposure to genomic epi tools. And finally, we'll talk about the epidemiologic questions that phylogenetic trees can help us answer. So I wanted to start off with this slide that really just helps us sort of set a stage or a framework for understanding how whole genome sequencing results can help us discriminate different levels of subtyping. So where we discussed in one of the first lessons how, the Carlos-Linnaeus system of kingdom, phylum, class, order, family, genus, species is still in effect, but now we can identify many different levels beneath the subspecies level. And so this diagram just sort of explains in terms of seasonal influenza viruses how we have been able to discriminate beneath that subtyping level, right? So if we just consider influenza A and B, for example, and then think about the different subtypes, 2 main seasonal subtypes that we see in the United States, H1N1 and H3N2, and then understanding how those are then further broken down into clades and then subclades. And then when you're looking at influenza B, we have lineage names that you may be familiar with in terms of Victoria or Yamagata for influenza B. And those can be broken down further as well by clades and subclades. And so really the point of this slide is just to think about, influenza in relationship to what you may already be familiar with from dealing with during the pandemic with all the different SARS-CoV-2 variants and subvariants and all the different names from Alpha, Delta, Omicron, et cetera. We really know a lot about the SARS-CoV-2 virus now at this point, 4 years after the pandemic. But when you think back to influenza, we have also been sequencing influenza since the 80s as well. And this is another virus that we know a lot about. And so again, whole genome sequencing allows us to discriminate at much lower levels beneath the subspecies. And all of these are based on SNPs. And that is really the take-home point here from this slide as we start to think about how to apply the results from whole genome sequencing. Okay, and so this slide also, shows that kingdom, phylum, class, order, family, genus, species, and how we can then break down even further. And so there's different ways of doing that, right? We've had other slides, I think, during Nick's presentation that talked about the different types of subtyping. And just to spell it out here, there's MLST, so the multi-locus sequence typing, as well as 2 different types of MLST. So whole genome MLST versus core genome MLST. And so again, similar to when we were talking about WGS versus NGS, right? Whole genome MLST just looks at the entire genome when trying to determine which sequence type and to make those calls when discriminating and classifying a specific sequence. Core genome MLST looks at core genes. And so when I think about core genome MLST, I always like to describe this as, thinking about the body or the body systems, right? So humans have, a brain, heart, lungs. These are main organs that are required for maintenance of our bodies. So core genome or core genes that are used for core genome MLST, often bacteria have 7, and these core genes are those genes absolutely required for maintenance of that bacteria to keep it living. And so again, this slide just really shows, how we can use those different subtyping methods to discriminate far beneath the subspecies level. And all of these are ultimately based on those single nucleotide polymorphisms or the lowest level of mutational change in the genome or the whole genome sequence of a specific pathogen. Okay, so with that context, right, as we continue to think about what a SNP is and what it really means, we need to understand what we would count as a SNP difference. So I like this slide just because often when we think about SNP versus allele, sometimes for epidemiologists, there's a little bit of confusion. So if you think about that SNP being a single nucleotide base pair change, if a SNP occurs in a gene, then that gene is referred to as having more than one allele. And so you can see on the left where it's showing this intergenic region or a gene in which you see a G change to a C nucleotide base pair. But when you look at the right, if this gene has multiple different SNP changes, then this gene would be considered to have 3 different alleles. And so just looking at this side by side helps me think about, when you see PulseNet thresholds for differences between enteric bacteria, when they say, these differences are based on allelic differences, it is really actually different than saying these are SNP differences. So there's definitely a distinction, and sometimes it's used interchangeably when it should not be. So I just wanted to make that clear distinction here in the beginning. Okay, so for the where to call a SNP, or where is it, SNPs basically are called based on, a couple different factors in terms of looking at the sequence data. So there's different quality scores that are calculated as a part of whole genome sequencing, and a lot of this information is sped off of the MiSeq sequencers themselves, in terms of understanding like the Phred score and the coverage and the quality and understanding base frequency. But all of these things, even if you're not a bioinformatician and this is not the main thing that you're looking at every day, understanding these concepts is helpful for understanding when and where to call a SNP. So again, the differences between the reference and a compared genome are extracted and then used to determine how related they are. So you're going to compare these sequences to that reference sequence. But not all pipelines are equal. Where you call the SNP will affect that total SNP count. And depending on where you're looking for SNPs, you only want to call a SNP in areas in which you have really good quality, right? So this is just a nice figure that shows where to call SNPs in genes. So these would be, for example, like thinking about allelic differences in one particular gene if there's multiple different changes. So like 2 slides ago. And then this also shows areas here in the center where there isn't good coverage, where you may not want to necessarily, look for a SNP in that area. And there's also an area on here that talks about masking of mobile elements. So this is not a good place to consider calling a SNP in that location either. So again, just try to understand high level in terms of this concept. And then this slide shows you what a multi-sequence alignment would look like. So this is where a bioinformatician may be comparing all of these sequences side by side, and where you can see it's a little bit easier to see when there is a single nucleotide polymorphism change. Okay, so when we're thinking about SNP matrices, this is a really pretty basic slide, but I just wanted to make sure, again, that we're all on the same page in terms of thinking about what a single nucleotide polymorphism matrix is, right? So this is literally 2x2 comparison of, you list all of the isolates on one axis and the same ones on the other axis, and we're literally comparing them 2x2 in pairs in terms of how many single nucleotide polymorphisms difference are there between those. So in this specific SNP matrix that is visualized here, all the vertical gray boxes here represent when one single sequence is compared to itself. So there should be 0 SNP differences between one and the same. Some SNP matrices actually list zeros instead of these gray boxes. And so it's important to understand that there can be 0 single nucleotide polymorphisms between, say, this WGSID 3 and 1. So where in this matrix it says 3, but this could be a 0. And so it's important to pay attention and look closely at the SNP matrix that's provided to you. All right, so just with that orientation to make sure that you understand how a SNP matrix is represented, often SNP matrices are called heat maps, and you can add heat or color to denote certain threshold differences just to look at the SNP matrix a little bit more easily. And so often, you see, like, say, this one is probably showing 0 to 5 as light yellow, and orange would be, say, 10 to 15, and anything more than 15 is then colored red in the specific SNP matrix. So that also just helps the eye as you're looking at it. So again, what inferences can we make from a SNP matrix, and what can we not see from it? So essentially, these tables are useful in determining relatedness between isolates. It's a digestible format, but it cannot show us the evolutionary relationship between isolates. Sometimes they can show clustering of samples, but they can't necessarily show us how these clusters of isolates are related to each other. I think it's better. Yeah, it's better explained on this next slide. So in terms of looking at the phylogenetic tree in comparison to the SNP matrix. So in the first lesson, I mentioned that often we will get a phylogenetic tree without a SNP matrix. Sometimes we'll get a SNP matrix without a phylogenetic tree, and that is not ideal. Ideally, we would want to have both of these so that we could compare them side by side to better understand, again, like what are some of these isolate numbers that are thrown on here? What are all the other annotations, and hopefully the phylogenetic tree that we get is decorated with some additional details like this one. And that allows us to, again, better understand, make inferences, and then apply these genomic results to the investigation or surveillance data that we're looking at as epidemiologists. So again, in this particular SNP matrix, you can see from 0 to, say, maybe 25 are listed here with bright red, but these are obviously much more closely related than, say, the ones in light pink that are 25 and greater, and then the blue and the darker blue. So when you want to compare the SNP matrix directly to the phylogenetic tree, we can see here that these top 3 isolates are 1 to 2 SNPs apart, and then you can take your eye down and compare the ones ending in 903, 4, and 5, and you can find them on the SNP matrix and see that yes, they in fact do cluster here with 1 to 2 SNPs difference. The other cluster that are only 2 SNPs apart, the ones ending in 968 and 651, you can find here in this smaller cluster, 0 to 2 SNPs apart. Now, if we want to understand what is the genetic evolutionary relationship between these 2 clusters, then we need to look at the phylogenetic tree and compare how many SNP difference, between this cluster and the next one. All right, we're going to explain that a little bit more a little later. So I will pause there. But again, the take-home point here is that we want to compare both of them together. The SNP matrix cannot show us evolutionary relationships by itself, and the phylogenetic tree cannot infer directionality of transmission by itself either. But we want to try and look at this information together. Okay, and so here, how can we use the SNPs to build a phylogenetic tree? This is a toy diagram, if you will, just trying to, again, ensure that we're all on the same page and understand some of the fundamental concepts. But in reality, it is a little bit more complicated than this. But for this phylogenetic tree, it shows you an ancestor sequence. So this one here in the root of this phylogenetic tree. And when you try to compare this one to isolate D, for example, you can see that there are 5 single nucleotide polymorphisms difference between isolate D and the ancestor. And so you can essentially see that the x-axis here is showing genetic change over time in this genetic divergence tree. And we'll talk about what that means a little bit in a minute. And then you can compare how isolate D relates to isolates A, B, and C that are farther apart on this phylogenetic tree. Okay, so just a note about bootstrapping here for a minute. Bootstrapping, again, I mentioned that sometimes the tree, you'll get the phylogenetic tree by itself without any of that SNP matrix information. And so we don't really have a lot of good understanding here in terms of like SNP differences between all of these viruses. But sometimes you will get a SNP matrix that is, a phylogenetic tree that is labeled with these bootstrapping values. And this does help us understand a little bit about the way the tree was constructed. So bootstrapping are numbers that denote the statistical confidence in the shape of the tree or the topology. So for example, here where it shows virus 3 and 4 and there's 1.0 labeled, this is essentially saying if we were to draw this phylogenetic tree 100 different times, and we have 100% of those times, we have the highest confidence that virus 3 and 4 would fall in this location on the phylogenetic tree as being genetically related. But then, for example, back at this node, we have 0.65 for the statistical confidence that 65% of the time virus 3 and 4 would fall in this relationship to virus 1 and 2 in terms of them sharing a most recent common ancestor. So again, this is the measure of the tree topology happening by chance, versus reality. Okay, so this is, again, a oversimplification, but this is a sort of a Mickey Mouse phylogenetic tree that is just showing, the relationships between certain animals or organisms. And so phylogenetic trees are used to infer the shared and unique genetic history across a set of organisms or clinical samples at the same time. And so the development or evolution of a group of organisms or the evolutionary history of those group of organisms are depicted in this way, similar to, say, a human, genetic family tree, if you will. And so what's important to think about with these phylogenetic trees, if, in terms of where the nodes are located and how, the most common mammal ancestor would be here at this node, and meaning that this is the most recent common ancestor between the bear and the chimpanzee. And you can work back. So it's just important to pay attention to where the nodes are. Okay, so if phylogenetics is then this, it's examining this evolutionary relationships of organisms through their genetic material. So again, we are looking at those whole genome sequencing results, the single nucleotide polymorphisms. We are doing multi-sequence alignments to then compare what are those in fact differences. And so just to cover two main types of trees, you can have rooted versus unrooted. I tend to prefer rooted. I find unrooted a little bit difficult to conceptualize sometimes. But the rooted trees are showing an implied evolutionary relationship between all of these isolates that they share a most recent common ancestor, where an unrooted tree just shows the relationships between these organisms themselves, right? We're not comparing them to one common evolutionary source. Okay, so for anatomy of a phylogenetic tree, again, these are just, to make sure we're all using the same language and we understand some of the basic concepts. So, we already mentioned the root being the farthest point to the left on the phylogenetic tree in terms of the x-axis. And all of the isolates that are labeled on the very far right at the end of the branches are considered leaves. Nodes are when we're working back from those branches and we find a point that is in common. And what's important to note for phylogenetic trees is the scale. You want to pay attention to whether or not it is showing genetic divergence, so it would be a genetic divergence tree, or if time is labeled on the x-axis, and then that would be a time-based tree. And so we'll talk about those a little more in a minute. Okay, so topology, in terms of talking about the shape of the tree, it's really important here to understand that any of the branches can be rotated around the axis of the node. So these two phylogenetic trees show the exact same thing, even though the bottom branches are rotated around this node G. So these are in fact identical. And what's a little bit tricky about that is when you're not in control of generating the phylogenetic trees, or you're getting phylogenetic trees from bioinformaticians or from CDC, or whoever did the sequencing and bioinformatic analysis, sometimes the tree that you get handed looks very different. And in fact, they may be exactly the same. So it's really important to take a look at your phylogenetic trees when you get them, to be ensure you understand exactly how they're labeled, and then decorate it yourself if you can to make it make more sense to you. All right, and so this shows a tree that is standing in the forest. I like to look at the phylogenetic trees that are laying down in terms of having that root on the far left, but sometimes they can be depicted in this vertical orientation. And so again, it's important that you just pay attention to where those nodes fall as you're looking back on the branches. Okay, so this was just a test or a quiz to check understanding in person, but these are questions just to ensure that you understand. So for the first one, which is the most recent common ancestor for isolates I and D, you would then need to go back on the phylogenetic tree all the way to the root to find the most recent common ancestor for these two isolates. The most recent common ancestor for C and D would be A, and the most recent common ancestor for L, H, and K, you would need to trace all the way back to this node here, F. Okay, so strains that are phylogenetically closer are more likely to share an epidemiologic link. So this is really just the the premise behind a phylogenetic tree building or generating, and how we can then use the phylogenetic tree in conjunction with our epidemiologic interview data to confirm or refute certain hypotheses about genetic relatedness. All right, so for some tree building considerations, again, how we were talking about in previous lessons about how they're often very pathogen dependent, again, it's important when you're looking at whole genome sequencing results to think in the context of which pathogen you're talking about. Same for the source type, right? So if you're comparing isolates that are from different sources, whether it be clinical or environmental, that's an important aspect to remember. Time and place, right? So we already talked about how we can have time-dependent phylogenetic trees, labeling time on the x-axis. Place could also be in something that we take into context when building trees, and overlaying some of those geographic details. And then, again, we've already talked about how a phylogenetic tree cannot show directionality of transmission. And same goes in terms of thinking about how to apply the results from a phylogenetic tree. This cannot be used in isolation to solve an outbreak. Often, EPIs, we would like to think that we can solve the outbreak by ourselves, but, and maybe we can, but the laboratory data definitely adds a greater resolution to the investigational information that we have. And same goes for the laboratorians. They can't solve an outbreak by themselves without all of the additional epidemiologic metadata that really enhance the information that they've generated in terms of genetic relatedness. So all of those epi interview details, contact tracing, exposure information, any clinical features or information that we have about the actual disease severity or symptom onset about the specific disease. So all of these are just things to consider when designing the tree and when designing the analysis and determining which isolates to include in the phylogenetic analysis. Okay, in terms of methods for tree building, I'm not going to go into all of these in great detail. Often I consider some of the mathematical modeling behind phylogenetic trees a little bit like reading a mathematical textbook. For me personally, when I get to the sections with all the funny shapes and symbols and letter or numbers, I often push the I believe button and need to just accept the fact that the theory works and this is how it's, the outcome that we see. But in terms of the mathematical theory behind it, I'll jump down to the bottom just to say that, I can understand neighbor joining. This is really, the basis behind looking at the, SNP matrices and comparing isolates, 2 by 2, looking at the relationship between 2 and then adding a third. That would be, say, the neighbor. And are they, so basically starting with those pairwise comparisons and then adding a third. And then those are, again, the foundational understanding of the phylogenetic tree, that there, 2 would be rooted together with the most recent common ancestor, and you work your way back to, the primary ancestor for an entire phylogenetic tree. So the neighbor joining makes really good sense to me. Maximum likelihood, we do a lot of statistical analysis in epidemiology with that mathematical theory as well, and that makes pretty good sense to me as well. UPGMA, you can read for yourself. This means unweighted pair group method with an arithmetic mean. And this is, a little bit more complicated, but it is also using this concept of the distance, the distance between two differences between isolates based on their single nucleotide polymorphism changes. So what's important to take away from the slide is just that, no 2 trees are the same. And it's important when you get a phylogenetic tree to try to understand or get information on how it was built. What were the methods behind the tree? Because you cannot compare a phylogenetic tree that is generated using neighbor joining to one that was, created using maximum parsimony. They're just going to look different. So just like previous slides had stated that no 2 bioinformatic pipelines are going to be give one and the same outcome or results for us. Neither would, 2 different types of tree building methods. We've already mentioned reference versus reference free. This really refers to, again, are you referring back to one common reference strain, when doing your multi-sequence alignment. And this also goes back to thinking about sequencing, whether you're, cutting up the whole genome and then reassembling it based on a reference. Or are you doing de novo sequencing where you aren't using a reference as you're reassembling that genome? We already mentioned core genome versus whole genome. Again, point here just being that you don't want to compare apples and oranges. You want to compare, core genome MLST results with other core genome MLST results. You don't want to mix and match. And then in terms of methods for tree building, we mentioned that there can be time calibrated trees or temporal trees where the x-axis really denotes time and shows genetic relatedness between the isolates over a time scale versus genetic divergence, where we really are showing how they are related based on their single nucleotide polymorphism differences. Okay. So now just for a couple visualizations to really ensure that we have taken on board these concepts. So, we mentioned a little bit about in the first lesson about sampling design and ensuring that we get a representative sample of, say, our entire state if we're looking to do surveillance of one particular disease. And the reason for that is because we want to ensure that we have good representation and without, ongoing surveillance with contextual data that we can then compare, isolates that cluster together, it's difficult to determine if you see an increase in disease incidence compared to, the background expected level of incidence. Same for when looking at phylogenetic trees. Looking at a tree without contextual data, will give us one level of understanding. But when we add contextual sequences or additional data to that phylogenetic tree, it paints a different picture, right? And so often analyses are limited by what we have sampled. And so unfortunately, we can introduce a certain level of biasy based on how we do this sampling. And so the contextual data really does tries to reduce the amount of biases that we have when looking at phylogenetic trees. And again, these images came from the Genomic Epi handbook created by Ali Black. Oops. And I've also borrowed some slides from Trevor Bedford's group on NextStrain, so I just wanted to give that acknowledgement as well. So we've mentioned a couple different times that phylogenetic trees are not transmission diagrams, and so this diagram or this slide really just shows, if we were only to have, these isolates here that are in blue, again, it would show a different picture if this is what we have sampled and trying to understand, what is the point of transmission between different isolates on this tree. If we had more contextual data and we had, additional isolates that you can see here, the red and the blue, then we would have a different understanding of how, the disease might have spread. But phylogenetic trees, we cannot determine directionality of transmission. Okay, and this one just sort of drives that home even further. So the top shows these are genetic divergence trees showing nucleotide changes over time. And in the top diagram in the left panel here, we can see isolates 1, 2, and 3, so show 0 nucleotide differences between them. They're all identical, where the isolate number 4 has 1 nucleotide change. And so if you were to look at the bottom here, this one is representing a transmission diagram, right? Not a phylogenetic tree. And this is showing the true transmission pattern where individual 1 infected individual 2 and 3 and so on. We basically observed that there were no mutational changes between these transmission events between all of the first 3 patients here. But by the time there was a transmission event between patient 3 and 4, there was a change in that consensus genome and a mutation did occur. And now we see a genotype B. Again, these are toy diagrams, very oversimplified, but it's just trying to show that what the true transmission pattern would look like. But if you were to look at the panel here on the right, again, you see a very similar phylogenetic divergence tree generated where isolates 1, 2, and 3 are identical and 4 is only 1 SNP difference. But another way that this, could have actually happened in reality, it could have been a super spreader event where person 1 was the direct infector of all persons involved in this image and visualization. So, within host diversity was actually present in person 1, where this could be an example of this could be, say, a patient who has hepatitis C and they have a quasi-species infection where they're infected with more than one genotype A and B. And as they transmitted this hepatitis C to 4 different individuals, The minority variant was only transmitted to person 4, where the variant that was the majority in their infection actually was transmitted to 2 and 3. And so again, you can see that true transmission patterns may differ quite widely from the actual genetic divergence that can be represented in a phylogenetic tree. So again, this just further illustrates how you cannot infer transmission by looking at a phylogenetic tree. Okay, and so for the last part of this lesson, we just wanted to talk about genomic epi tools and understand that we do not, give an exhaustive list of all that are available. These are just a couple that we've focused on here, just to give some general exposure and to talk about these in terms of different categories. They can be organized based on the purpose or intent of the tool, and you may not work with any of these in your real day job. You may get the visualizations or results from some of these genomic epi tools that, say, a bioinformatician worked with or a genomic epidemiologist worked with. But we thought that it would be important to include in this lesson just sort of an overview of how of some of these tools and how they're utilized. So in general, they can be one main category would be tools that are used for data collection or metadata curation in some ways. And so a lot of you are already familiar with EpiInfo or REDCap surveys in terms of collecting data, but they can also be used in conjunction with R for statistical computing, and you can use the exports from these tools to do some basic metadata curation and visualization. In addition, genomic analysis or tree building tools, are tools that allow you to do some basic analyses or run some statistical models to create phylogenetic trees. And an example of those would be ITOL as well as Usher and NextStrain, and we'll talk. I'll show some screen grabs from these as well. And then there's genomic epi tools that only allow for visualization. So these would be maybe to show a network or a geographical representation, say on a map. And they can also do some aggregate analysis, but in general, they are not doing the mathematical modeling behind placing isolates on a phylogenetic tree. And so R can again be used for these visualizations, as I mentioned, NextStrain, MicrobeTrace, and MicroReact, and I'll show all of these. Okay, and so I often get questions about R. We have some epidemiologists in Virginia that are really skilled with R, more so than me, and I anticipated this question: can you make a phylogenetic tree in R? Well, of course you can. It is, it's difficult to actually decorate the tree itself in terms of changing the colors or what have you of these leaves on the phylogenetic tree branches, but somebody will probably correct me later and say that there's a way to do it. I just didn't know it. But I do like how you can then make a key for all the different color coding that you assign on the right-hand side here, alongside of your phylogenetic tree. So, it's not so hard to have your eye drift across and identify what these are, as long as you have a very clear key. And so the EpiR handbook has a really nice description of how to go about coding these visualizations in R. I prefer interactive tree of life in terms of being able to directly annotate the tree itself and change those the labels and colors and coatings of the branches and the labels of the leaves themselves. But there's a freeware version of ITOL, and then you can pay for a license to be able to save and then manipulate your trees a little bit easier. Usher, these slides I've borrowed directly from the CDC COVID-19 toolkit for epidemiologists. If you haven't yet checked that out, please take a look. There are many modules that explain a lot of some of the things that we've covered in these series in great detail. So Usher is literally this acronym, ultra fast sample placement on existing trees. And so Usher does allow you to do that. It does the mathematical modeling of placing the isolate directly on the tree itself. And it runs relatively quickly. And what's wonderful is that the Usher output then opens up into NextStrain, and then you can see it clearly in context with other contextual sequences that are already uploaded to NextStrain. And here is an example of NextStrain. Many of you have already seen screen grabs and builds from NextStrain. Our own bioinformatician, Logan Fink in Virginia, maintained a NextStrain build for the beginning of the pandemic. I don't know, maybe the first 9, 10, 12 months. And then, it just became untenable. But this is actually a screen grab of a CDC Spears build that is specific for Virginia. And these are not all of our sequences, obviously. These were just a focus sampling of certain isolates. But NextStrain really gives you a wonderful utility in that you can toggle back and forth between this type of tree, which is a time-based tree, to a phylogenetic divergence tree to show just those single nucleotide polymorphism differences on the x-axis. So I really like that feature in NextStrain. Okay, quickly, just to wrap up and showing a couple other tools that are available to you. Again, just to get exposure here, and each one of these tools have tutorials and lessons and a lot of videos on the MicrobeTrace website that CDC maintains to teach you how to use MicrobeTrace itself. But this is a really versatile tool that I've come to love. It is powerful, and in that it is secure. You don't have to upload your data to anywhere in the cloud. It operates, you download the program itself to your computer, and then you can run your analyses locally. And then just to quickly show, it allows you to build transmission network diagrams, and there's a bunch of different representations that it can allow you to display. So you can show person to person, like contact tracing we did during SARS-CoV-2. It can also show epi network in terms of exposures where a person, may have been exposed to a certain place, whether that be work or, a certain restaurant. And then it can also show person to place. In terms of, this is another example in terms of the place being their residence. So this could be like for SARS-CoV-2, household transmission, within the same household. But it's really helpful in terms of trying to understand the transmission that is going on. And you can use a genetic distance network when you have SNP matrices and genomic results available to show genetic distances between the viruses as well. So on this slide, you can see an integrated example, and essentially this shows the epimetadata overlaid onto the MicrobeTrace visualization. You can see nodes colored differently, and it allows for you to include a legend describing what all of those different annotations mean. And so our last genomic epi tool that we wanted to cover is MicroReact. And this is a really special tool in that it allows you to create dashboards and visualizations with multiple different panels inlaid. And what's really nice about it is that when you make any changes in one panel, say to display, certain SARS-CoV-2 variants in this example, then it will also make those reflected changes in all of the other panels at the same time. And so one of the other nice features is it offers an epi curve or a timeline that you can add for one of the subpanels, and allows you to see, cases over time. And it also allows you to create different views. So once you have the dashboard set in a certain way, to make the visualization, you can save those views and then share links to those views with other epidemiologists that you want to, see that visualization. So you don't necessarily have to have super good familiarization with the tool itself or even the data that's involved to share it with someone else. And this is just an example of one MicrobeTrace link that we received from CDC about Candida auris cases a long while back, but this just kind of gives you a zoomed in view, and you can see they had highlighted Virginia cases in green over here in this, the key on the far right. And so we could see where our cases fell against, the contextual data from other states that CDC provided to us. Again, all de-identified data, but it allowed for us to, again, just get a link and see this visualization, even though we didn't have all of the data sources and data sets that went into creating this phylogenetic tree. Okay, so just to wrap up this lesson, I wanted to cover, again, what are some of the epidemiologic questions that we can answer using phylogenetic trees and genomic epi visualizations. And so as in the first lesson where we were talking about how it's really important to know what questions we are asking prior to making inferences and drawing conclusions from genomic epigenomic results, it's important to kind of think about how we can go at answering these questions. So phylogenetic trees can be used to answer questions about time. When we're looking at time divergence trees, especially, this is useful. So we could answer questions about, say, thinking in the context of SARS-CoV-2, when a new variant or lineage was introduced into the county in which we're living or working in, or interrogating, and then how long has that lineage been circulating, or when did that lineage become associated or introduced into a certain outbreak setting, for example, a congregate living facility, or was it in the congregate living facility to begin with and then spread out to the community? These are questions that we can attempt to answer based on how we create the phylogenetic tree and do the bioinformatic analysis, right? So thinking about which sequences we're going to include in that analysis beforehand can help us answer these questions. So again, some of these questions are better to have sort of framed a priori before we start that analysis. Place. So we talked about using phylogeographical or spatial dynamics to attempt to understand, how the pathogen is spreading among places. And so often we can look at certain regions or counties and, throughout our states to, try to draw inferences about how that variant is moving about. And so this, leads to this understanding of transmission dynamics. So again, we cannot infer direct transmission directionality from a phylogenetic tree, but we can try to understand, a little bit of the larger picture, especially when we have ongoing genomic surveillance as contextual data that we can include on that phylogenetic tree, right? So does our region tend to act as a source or a sink for transmission? Is this pathogen coming in from outside locations, or is it starting from within our region or county district? Is our outbreak driven by frequent introductions with limited transmission, right? So are we seeing new variants appear and then we're not seeing sustained transmission? And then, did our public health intervention slow or change transmission? This is one of the ultimate questions we're always trying to understand, especially, with some of the healthcare associated infection investigations, again, where we're doing that bottom-up approach, where we really are trying to contain the disease and prevent additional cases or from the particular pathogen spreading between, units in a facility or between facilities themselves. And so are the recommendations, the infection control recommendations that we've made and put into place, are those actually working to slow or change the way the pathogen is being transmitted? So again, just food for thought here as we start to, really move into the stage of understanding genomic results and working as genomic epidemiologists as to how we can take this information and apply it to the outbreaks that we are working, or, the clusters that we're detecting as a result of ongoing surveillance, genomic surveillance. And finally, I just wanted to include this acknowledgement slide. As I mentioned in the first lesson and throughout this one, we have borrowed a lot of content from multiple different contributors. Definitely a big thanks to Allie Black from Washington Department of Health, as well as several CDC sources in which we have cited from. Also, previous presentations that were included in CSTE and the AMD workshops. So thank you again to all those that have contributed to the training materials, and then who have participated in training a lot of Virginia Department of Health staff throughout the state. We've done 5 of these trainings in 2024. So we hope that you've enjoyed these series and that you have benefited greatly from these. Thanks.